enzymes has a defined three-dimensional structure of amino acid sequence. Enzymes are big in size compared to substrates which are relatively smaller. When you take an enzyme, the entire enzyme molecule is not very active. There is a small site in the enzyme which is responsible for this catalysis. That small part is known as the active site. So what exactly is this active site? Active site is nothing but active site of an enzyme is defined as a small region in which the substrate binds and participates in catalysis. Hence, there are some special features of this active site. So, let's move into the salient features of the active site. Firstly, the active site is formed due to the tertiary structure of protein resulting in a three-dimensional structure. This active site is made up of amino acids which are far from each other in the linear sequence of amino acids which are called as catalytic residues. Out of the 20 amino acids that could be present in the enzyme structure, only some of them like aspartate, serine, cysteine, histidine, lysine, arginine, glutamine, tyrosine, etc. are found in the active site. The active site is very flexible, that is to promote the substrate binding. The active site possesses two sites, that is substrate binding site and a catalytic site. The substrate binding site is a site to which the substrate comes and binds, and catalytic site helps in catalysis. The active site is like a cleft or a crevice. So here in the cleft or the crevice, the substrate comes and binds and the reaction takes place. When the substrate comes and binds to the active site of an enzyme, there is a formation of an enzyme substrate complex which leads to the product, and the enzyme is regenerated as it is. How do enzymes act? Let's go inside the mechanism of enzyme action. For any chemical reaction to occur, the reactants have to be activated state or in the transition state. Let's see the activation energy. The energy required by the reactants to undergo the reaction is known as activation energy. So here you can see in the graph regarding the activation energy. The reactants, when heated, attain the activation energy. The enzyme reduces the activation energy and this causes the reaction to proceed at a lower temperature. The enzyme lowers the energy barrier of reactants, thereby making the reaction go faster. How do enzymes act? Enzymes speed up the various biochemical reactions, that is, they accelerate the velocity of the reaction. The prime function of the enzyme is catalysis. Let's see the mechanism in detail. So, the enzyme catalyzes the reaction in two steps. That is, the first step is it lowers the activation energy, and the second step is it forms the enzyme substrate complex. Divulge detail about the lowering of the activation energy. So, for any chemical reaction to occur, the reactants has to be in a activated state or a transition state. The energy which is required by the reactants to undergo the reaction is known as the activation energy. So here this catalyst, that is the enzyme, it reduces the activation energy which causes the reaction to proceed at a lower temperature. It lowers the energy barrier of reactants, thus making the reaction to go faster. As a result, all the reactions in the biological system occur at body temperature. Hence, enzymes do not alter the equilibrium reactions, they only enhance the velocity of the reaction. Next, we will move on to the enzyme substrate complex. What is this enzyme substrate complex? So, the primary requirement in a reaction is the enzyme and the substrate. The enzyme reacts with the substrate to form an ES complex, which is the enzyme substrate complex, which will give rise to the product. So, there are very few theories which is put forth to explain the mechanism of the enzyme substrate complex. We have the lock and key model, Koshland's induced fit model and the substrate strain theory. First, let's move on to the lock and key model. So, this lock and key model was proposed by Emil Fischer in the year 1980. Just as how a key goes and fits into a lock, the substrate goes and fits into the enzyme molecule. This theory was not very much popular. Why? Because here both the enzyme and the substrate is very much rigid and it is not flexible. The substrate fits into the binding site of the enzyme just as how a key goes and fits into the lock.